Hello, everybody. So welcome to our first lecture of Chapter 3, which is going to be on cell membranes. So let's get right to it. Now, by the end of this module, you should be able to discuss the structural and functional features of the prokaryotic plasma membranes and be able to compare bacterial and archaea plasma membranes. So here we're just going to talk about the prokaryotes. All right, so we call the outside of the cell, which includes the membrane and the cell wall and the periplasmic space, the cell envelope. And basically the function of the cell envelope is to separate the inside of the cell, where your cytoplasm is, from the outside of the cell, all right? And you're going to have these three parts: the plasma membrane, the cell wall, and the periplasm. We're going to go kind of in depth about the plasma membrane and the cell wall. The periplasm we're not going to talk as much about, but there will be a little bit filled in here and there. And so that cell membrane or that cell envelope acts as a barrier, right? And in this case, we've got our plasma membrane, and this is going to separate from the intracellular stuff. So everything in here is considered intracellular. From the extracellular stuff, everything out here is extracellular, right? And so, for example, this flagella would be an extracellular structure. This chromosome would be an intracellular structure, right? Most of our prokaryotes are going to have a cell wall, and that is going to be outside of the plasma membrane for gram-positives, and for gram-negatives, it's going to actually be between the two plasma membranes. But we'll talk more about that with the cell wall lecture. Now, the plasma membrane needs to be flexible, and it is composed of a phospholipid bilayer. So we learned a little bit about phospholipids in the last chapter. So we've got these hydrophilic heads and these hydrophobic tails that form into this bilayer. And this is fairly thin. And it's going to selectively keep some things inside the cell and some things outside of the cell. So it's a selective barrier. Some things can get through and some things need permission. And so here's just a reminder of what those phospholipids look like. We've got our bacterial phospholipids on the left-hand side, and then our archaeal phospholipids on the right-hand side. We've got that phosphate glycerol combo up here, which is going to make it hydrophilic, and then we've got our fatty acids down here, which are going to be hydrophobic. And so, again, this is your interface between the inside and the outside of the cell. And that's going to determine how the cell functions. Now, something kind of interesting about these phospholipid bilayers is that the proteins can actually constitute up to half of the plasma membrane mass. So it's not just phospholipids, it's actually going to have all sorts of proteins through there. So we're going to have things that span from extracellular to intracellular, like this guy here or this guy here. And these are going to be like a transmembrane, so trans meaning across protein, so a transmembrane protein. And then we're going to have peripheral proteins. You can have peripheral proteins intracellularly, and you can have peripheral proteins extracellularly. And so those peripheral proteins don't span the entire membrane. Now, the proteins that we find in these plasma membranes can have multiple functions. For example, we have receptors like these TKRs and GPCRs that allow for cell-to-cell communication. They can sense what's going on in the environment. These receptors, for example, might indicate what direction to go in chemotaxis. Now we have cell adhesion proteins like these over here. These allow your cell to adhere to the surface of other cells or to other surfaces. These cell adhesion proteins, when they adhere to other cells, are going to be fairly specific to certain types of cells usually. We've got enzymes, which are going to be the things that they're types of proteins that catalyze reactions. And so you have some of these that are released extracellularly. You also have some of them that sit in the membrane, and their job is to break down complex molecules, usually into smaller subunits, to allow them to enter the cell. Then we have transporters. These move substances in and out of the cell that can't pass through by a simple diffusion. So we're going to talk a little bit more about transporters in a separate lecture. So the plasma membranes exhibits selective permeability, and that means that some things can just go straight through. They can permeate the membrane. These are gases, water, and small non-charged or non-polar substances. They can go right on through, right? So CO2, O2, those sorts of things can diffuse through the membrane. However, ions and larger polar substances need to have some sort of transport protein. And again, we're going to talk a little bit about this transport more, but you have channel proteins or carrier proteins, and they basically function to help these other things in and out of the cell when needed. Many of your membrane proteins can detect changes to the environment. And so, as I was mentioning before, you might have a receptor like this right here, which comes into contact with, say, a sugar molecule, and then basically encourages that cell to go towards a more concentrated gradient of that molecule. So this is a combination of a receptor protein that's going to find this signal, and then a motor protein that's going to help move towards that signal through a process called chemotaxis. And in this case, it looks like they're using polar flagella. Now, membranes are built in, or at least the way that we think of the lipid bilayer, is a fluid mosaic. This means that you're going to have all these lipids and all these proteins in there, and they can move around. And that fluidity is essential to function. It allows you to insert more proteins when needed, remove proteins that aren't necessary, translocate proteins that need to be in a different spot. Now, this fluidity is important for physiology, and it's going to be largely determined by the type of phospholipid fatty acids that are in that membrane, right? So your saturated fatty acids up here, if you remember, the saturated fatty acids have no double bonds, and they can be tightly packed. This is like your butter and, baking grease, stuff like that, lard. And then you have your unsaturated fatty acids, which are going to have these kinks. And those kinks are created by these double bonds. And those double bonds help keep the membrane from being so tightly packed together, which can allow a protein to come insert itself in here and then be moved around. Now, the fluidity can be affected by things other than simply talking about the saturation level of your fatty acids. Temperature can affect fluidity. So in this case, as you heat it up, you actually get this, what's called a liquid crystal alignment, where you get these changes in how these fatty acids align to each other. That increases your fluidity. When you are in colder temperatures, it's going to reduce your fluidity. And so coming back to look at these phospholipid fatty acids, they're amphipathic, which we talked about, or amphipathic, which we talked about in the lipids lecture. That means that there's a hydrophilic end and then a hydrophobic end. Now, there are a couple of differences between bacterial and archaeal phospholipids, one of which is that generally we have these linear fatty acids, right? They might get kinks in them, because of double bonds, but bacteria just have these linear fatty acid chains. While your archaeal fatty acids generally have these branched lipids, so you get these little isoprenes that shoot off from the fatty acid chain. And some archaea actually have a monolayer, and the way that works is that they have a phosphate group up here. They have this phosphate head, and then down here they have another phosphate head. So they still have their they still have their fatty acid chains, but they're connected by two phosphate heads. You can kind of see that in this down here. So it's a monolayer because it's only one set of fatty acids instead of being a bilayer like this where you have two sets. And then another major difference is the type of bonding between that head and the fatty acid change. In your bacteria, you have ester bonds or ester linkages, right? So we have this COC. And then over here, we have our ether bonds, right? So these are a little bit different. Sorry, this is COC double bonded to O, and then this is just COC. And so I'm not super worried about you knowing the organic chemistry of this, more just to be aware that there are some actual differences between bacteria and archaea. These are some of the things that help us identify them as different domains of life. Now, some of our antibiotics are going to actually interfere with the plasma membrane. In this case, we're looking over here at these antibiotics like the polymyxins and the lipopeptide, easy for me to say, the lipopeptides. Woo, got through it. So these oftentimes are going to form pores in the membrane themselves. And so that's how they function and causing them to kill different organisms. I think for these, they're generally more effective against gram-negatives because they have this outer membrane. All right, so that is the plasma membrane of prokaryotes. So you should be able to discuss some of those structural and functional features of the membrane, as well as comparing the bacterial and archaeal plasma membranes. With that, there should be an objective check in D2L to see how you're doing with these two objectives, and I hope to see you soon. Bye-bye.